So we're live with Dr. Dan Stern Cardinale and Russel Empiricist, who we will call Andrew. I might call you Rat, I'm not sure. And we have Scott here, and we're going to have a conversation about cool science. And all you hyenas, all the rest of you hyenas, my wild band of hyenas, we're going to have Saturday Night Live at the Atheist, and we're going to keep this hangout to just the four of us. So that's what we're doing. Fair enough? And so, Andrew, you can go ahead and tell us what you would like to start us out with.
All right. Again, hold on.
That's putting you on a spot. I'm sorry, but that
is it. I heard an echo for some reason. Is it still echoing?
Not for me.
Huh? It was echoing on my part for some reason. I could hear my voice.
I heard it too, but it's fine now.
Okay, that's good. So first of all, I just wanted to say thanks for letting us chat on this subject, Dr. Dan. It's always a pleasure to have you on and hear your thoughts and insights on these kinds of topics. So I want to keep this focused on the process of science and how it hasn't always worked, or at least hasn't always worked the way everyone intended. And so I've divided these quick questions into 4 different parts, and so we'll go by each part and ask the questions in that part, and we'll go from there. So should I start with the first part?
Go for it.
All right. So part 1, infancy, how the scientific method was created. So I've put a little bit of history of science in here as well. And so my first question for you, Dr. Dan, is what is the difference between induction and deduction?
So it's the directionality of the reasoning, basically. Are you taking a big picture and applying it to specific cases, or are you taking specific cases and generalizing from that? And in science, you kind of need to do both, but different aspects of science are going to involve different sides of that. So you can take a broad idea and say, okay, if this idea is true, then we are going to make predictions and say, okay, X, Y, Z must happen under conditions A, B, C. And we can also say, we go the other way. We could say, oh, we've got this observation, that observation. We can generalize that to a rule about how things work. So you can go both ways there.
Right.
So you're talking about like on the timeline, both ways?
Kind of in terms of the directionality of the way the thinking works. You can either start with a broad idea and make specific predictions from that, or you can start with a series of specific observations and draw from that a broad, generalized idea.
Okay. Cool.
So Dr. Dan, you would say that both deduction and induction is useful is used in science.
Mm-hmm.
Okay. Would you say one of them? Sure.
Where does the idea of abduction come in here? Because I hear one of our creationist folks using that a lot.
Say that again. Adduction?
Adduction, something to the best explanation.
Yeah, abduction, which is
abduction with a BD?
Yeah, as in abduct or kidnap. Kidnapping science.
I'm not familiar with that one. Sorry.
So I'm hearing as a scientist you can live without it.
I think so.
Well, I mean, I'm not familiar with the term. That doesn't mean I've never used the reasoning, but I'm admittedly weak on the philosophy side of things, so me not knowing the term is not evidence that it is not relevant.
Very astute. Very astute.
There's a 2019 film called Abduction and a 2011 film called Abduction.
It's probably a common name for things. Yeah.
Okay. Not so important.
So
Dr. I don't want to say it's not important. I just don't know the term.
Yeah, right.
So I want to.
Right. So Dr. Dan, would you say that one was kind of used first, I guess, or like one was generally looked upon more optimally, I guess, in the early history of science?
That's a good question. I don't actually know the answer. The modern scientific method goes back a few hundred years to, as with any kind of thing like this, there's never going to be like one person you could point to that says like, aha, this person came up with this process in total. But to the extent that you can do that with anybody, it really comes down to Francis Bacon. I don't want to put a year on that because I just don't know off the top of my head.
Yeah, I don't know.
But I don't know enough about the history to tell you which one, if there was one that was earlier or taken from. I'm sorry, I don't know the history well enough to tell you.
Okay.
Is that the way I had it? I looked at it and I said, no, that's wrong. And then, so deduct is basically to reason, right? Reason to some model or whatever. So, okay. And abduct is, I forgot to spell things wrong. So abduct is like what is probable or whatever. And I guess creationists have been using abduction since 1883, right? Abduction, been using it to mislead science since 1983.
YEC, misdirecting the public since 1883.
Right. Yeah.
So is that going to be? So is that going to be like? Was I honestly don't know like how that term applies in this context, but is that going to be something like the big scary numbers arguments, the probability based arguments that like
because what
how could you have this stuff happen?
This ain't going to happen. This is too hard to happen. So therefore, the best explanation is this one belief that X% of scientists have who don't really do science anymore.
Well, you could make valid guesses about something with probability, like phylogeny is kind of a probability game. So that's kind of a little abductive reasoning there. Oh yeah, and stuff, and but for the most part, dividing reasoning into three classes and giving them names, I'm not a fan.
Right.
Let's just do the work. Okay.
So I guess so this wasn't really one of my questions, but I think it's relevant. Dr. Dan, do you have, I guess, a not a catch-all, but kind of like a run-of-the-mill kind of summary of what science is, at least essentially?
Ooh, put him on the spot.
Yeah, good question. So I always describe science first and foremost as a process. Right? It's not a series of beliefs. It's not a litany of facts. Science is a process. It's a process. It's really a process of elimination where you are proposing, testing, and eliminating explanations for specific observations, right? You want to know why a thing happens the way it does, come up with ideas of how that happens, and then you test those ideas through inductive reasoning. And in doing so, you eliminate potential explanations as valid by showing that they lead to incorrect predictions, right? So the way the logic flows is you make a proposal, that's your hypothesis, your proposed explanation, and then you make predictions based on that hypothesis, predictions that must be accurate if your hypothesis is correct. And then you test those predictions, and if they are wrong, then your hypothesis is wrong and you need to discard or change that hypothesis, right? So what we're doing is we start off with an open question and we are proposing explanations to explain that. And then we are eliminating those explanations through observations and experiments. And in doing so, we're converging on the most likely, the best explanation for whatever it is, the best answer to whatever question we're asking. So it's, at the end of the day, that process is what science is. And then I should say there's the next step in that is once you do that a bunch, you can then draw broader ideas from whatever it is you've observed and come up with. You can then draw, broader conclusions. You can come up with theories that are more general in nature and govern a wide range of observations.
Right.
But the short answer is that it's a process for evaluating the validity of proposed explanations.
Yeah, and you'd use these three things, I think, as part of that process to narrow down and arrive. And also, some of this stuff feeds back in and creates a new hypothesis, right?
So, absolutely. Yeah. When I do this in class, I show a big flow chart and we go through all the different things. And then at the end, everyone's kind of expecting a ta-da, you're done. But that's not what it is. Now it's go back and start again at the end. It's an iterative process that you never finish.
Right. Yeah, that's a good point. I think fundamentally we would agree that science is it's kind of, in a sense, it's finding patterns and using those patterns in nature to test our hypotheses, like you said. So like it kind of depends on the size of the quantitative or qualitative relationship we're looking at, and how broad that pattern is, and how many phenomena it affects in the natural world. Would you agree?
Sorry, say that again. I was just got an email from work at that exact moment. I apologize.
Okay. Yeah, I guess I can maybe rephrase it a better way, actually. So like we have, of course, patterns on the small scale where like we see, okay, if I drop this mouse over here, this computer mouse, this mouse over here, that's going to fall to the floor. So that's one pattern. And then another pattern would be, okay, if I drop this mouse and also drop these headphones and also drop this book, etc., they will all fall. So it's kind of, in a sense, looking for larger and larger patterns and trying to explain them as best we can to explain more and more of the observations we see. It's kind of connecting the dots to make a larger and larger image, right?
Mm-hmm. Yeah, and that there's a famous quote, and people are going to get on my case, rightfully so, if I get the person wrong, but I think it was Stephen J. Gould. But I'm sure there will be multiple people in the chat that will correct me if I'm wrong, if I have it wrong. But I think it was Stephen J. Gould, is he said, in science, the things that we agree upon that are facts. It's not that we've proven them. It's that they are well supported enough that it would be unreasonable to withhold your provisional consent. Say, like, based on what we know now, I accept this thing as true. Right? It's not proven. It's not going to necessarily be that way tomorrow, but based on what we know in the here and now, here are the things we can say with reasonable certainty are true, right? And science is the process of figuring that out. And right. I believe that same quote goes on to say, I could suppose that apples will begin to rise tomorrow rather than fall, but that idea does not merit serious consideration.
Right. And I think that brings up another good point, that theories, for instance, they don't really get overturned so much as refined and kind of incorporated into bigger and bigger models, would you say?
In general, go ahead, Speed.
Oh, that would be I think my systems patterns on the right, and I'm looking at, like, we had an earlier discussion about reduction and construction, and basically constructive is kind of what you're talking about, Andrew, with creating these greater patterns and systems. And I don't know. I hope my doodling and my craziness doesn't take us too far off topic. So proceed.
No, I think it's nice. Yeah, what I was going to say is I think that's true for theories that are robust. So, like, for example, you have ideas that are now completely discarded that we called theories, but they weren't. They were a lot of them predate modern science. So things like geocentrism or spontaneous generation, like these predate kind of the modern scientific method. And once they were evaluated using modern tools, and by modern I mean the 1800s, right?
Right.
Or for geocentrism, the 1600s, they start to not work as well. But for newer theories that came about in kind of the modern era of science, going back a couple hundred years, I think it's true that generally, rather than being completely discarded when we get new information, they are going to be refined. And physics is a really good example of that, where you've got Newtonian physics, and then it's not that that's wrong, but it stops working at certain scales, and relativity starts becoming a better explanation. And then that stops working at certain scales, right? You have quantum physics that becomes a better way of describing what's actually going on and why. Evolution is the same way. It's not that Darwin was wrong so much as Darwin just only got a little bit of the picture. And then since
Darwin
then,
wasn't
we've been building
omnipotent.
on. What was that?
I said Darwin wasn't omnipotent, basically.
Right. He proposed what turned out to be one mechanism of evolutionary change. I think it's funny that Alfred Russel Wallace, who was kind of the other person who presented with Darwin to the Linnaean Society in 1858, and was the first time anyone had proposed evolution by natural selection, Wallace developed the same ideas as Darwin completely independently, with one major difference, is that Wallace, in the letters that they both presented in 1858, and Origin of Species was published the next year in 1859. Darwin was like ready to go. He just needed a nudge, and Wallace coming up with the same ideas was the nudge. But in those letters to the Linnaean Society, Wallace actually described neutral theory. In his letter, he actually says, and by the way, what we're talking about with traits that help with survival and reproduction, that's actually not going to be most traits, and most traits aren't going to affect things enough to be subject to natural selection, and they're just going to be random. And it's one sentence, but he basically describes neutral theory a century before it becomes formalized.
Really? I didn't know that. That's cool.
In 1858, Alfred Russel Wallace described, it was literally one sentence in like a 3-page thing, but he describes neutral theory. But that wasn't incorporated in any formal way into evolutionary theory until the middle of the 20th century. And it wasn't really, really concrete until the work of Kimura and others in the 70s into the 80s. So that's a good example of where the theory, it's not that we're overturning an old idea, it's that we're adding to it. And the old ideas were not wrong, they were just incomplete.
Right. Right. Okay. So I think that's a good segue to go into the next part of the discussion, if that's okay with you guys.
Yep.
So this is part 2, conflict. So this is how science and religion sometimes wage war. So the first question I wrote down was, how often do scientists base their normally evidence-based conclusions on religion and or faith?
How often so faith is an interesting word there, because you could, if you interpret the word faith broadly, to include not just supernatural or religious ideas, but in general ideas that are held for emotional rather than evidence reasons, the answer is kind of a lot, it turns out. And it is hard to dislodge those ideas. And I'll give you a relatively recent example is let's see. We'll go with prions is a good example of this. So this was so prions are proteins that are misfolded. They cause disease. And the funky thing with prions is that they are able to, if a prion encounters the properly folded version of that protein, it will misfold it into the prion. So it's not self-replicating, but it can propagate the misfolding through a population of proteins of the same type, right? So this is causes like mad cow disease, for example. I forget the technical name for it. Something encephalopathy. I forget what the technical name for it, but it's mad cow disease, and there's a bunch of other diseases that do the same thing basically, and they're prion diseases. And I want to say it was the early 80s was the first time anyone proposed these are actually protein driven, and there's no genetic, as in DNA, basis for them. They are just purely based on protein folding, and the misfolded proteins are doing the work. And at the time, there was no data that specifically said that couldn't be the case. It had just never been observed before. It had never been proposed before. But there was nothing that said that cannot happen. And there was reason to believe, like, proteins affect protein folding. There's a whole group of proteins called chaperonins that help proteins fold. So is it crazy to think that a misfolded protein could fold other proteins into the wrong shape? No, there's no reason to exclude that out of hand. But it was basically excluded out of hand because, of course, proteins can't propagate themselves like that. You need an underlying genetic basis for that. That's silly. And it took about 10 years for that idea to gain acceptance, fighting tooth and nail. But by the end of the 1980s, it was accepted that prion diseases were due specifically to the proteins, and there wasn't like an actual DNA component to those diseases. I think if you use an expansive definition of faith, I think it was held on faith that proteins couldn't do that. And it took longer than it should have for it to be generally accepted that they could.
Isn't that like proteins? Basically, the prions, don't the proteins form globs? They kind of misfold and they end up with, and they kind of muck up the machine a little bit. Once they're misfolded, they cause all sorts of problems because they interact with things they shouldn't be interacting with. They can clump up with each other and gunk up the works. The really interesting thing about them, though, is it's the misfolded proteins propagate. So as the body goes, oh, we're running out of protein X, we need more of protein X to do whatever job, and you make more of that protein, the misfolded ones grab onto the new ones and go, now you're misfolded too, right? And that's not supposed to be able to happen, but it does. Chaperonins are kind of like, my understanding about the heat shock proteins, I think, are kind of like they're big alpha helices and beta sheet bags where the protein gets inside, and it kind of isolates. Isolates them long enough so that they can fold up properly, and then spits them out or something like that. That is my limited understanding of chaperonins. So
that's basically the idea. Yeah.
Okay.
So I would so if that kind of faith plays a role in scientific conclusions, does more religious faith play a role, or is that less common?
Well, go ahead.
Let's say, as a group, scientists tend to be less religious than other professions. That's not to say they're not majority. Most scientists are religious in some way. I believe that's the case. But as a group, it's less than other professions. And I think, part of the idea of science is the point of the process of science is to separate any one person's preconceptions and biases from the conclusions that you're drawing.
Right.
So you'll hear, like, I'll give you an example that I disagree with to tell you where I'm going with this. You're a creationist, say, for example, that it took so long to realize that more of the genome was functional than we thought of when we sequenced the human genome and realized, it's like 1.5% coding, right? Creationists will say it took longer than it should have to realize more of that non-coding stuff was functional because we had this preconceived notion that it was all random, it was all undirected, and there was no purpose behind it. But creationists were able to say, no, this is designed, this was created, whatever, therefore there should be some function in there. And therefore, creationists were like that faith background led to scientific discoveries, whereas the lack of that background actively hindered it. That's not true. That's not true. Like, it's just not. That's simply not true. And like the history of things like the ENCODE project show that that's not true. you can follow the idea, the history of ideas, like having a more than 1.5% functional genome, and it doesn't, that's not an idea that came out of creation science in the 70s and 80s or the intelligent design movement in the 90s. That idea, like, we knew non-coding parts of the genome were functional well before then. And then the specifics of it started to come into light once we could start sequencing genomes in the 80s, and then in particular by the late 90s when we got really fast at it, right? Like, so that's my take on that one.
Yeah, I don't remember anybody. I'd never heard the term junk DNA until I started listening to Standing in Poop. That was the first time I'd ever heard junk DNA. It wasn't, and I looked at all my textbooks, and they mentioned it in one little paragraph. Like they mentioned, oh yeah, that was an idea that somebody came up with. Somebody said it out loud at a press conference, kind of like the hard problem of consciousness. Somebody said it out loud, and it forever burdened us, right? And people can't let go of it.
But wait a minute. Isn't there a committee of evolutionists that are instructing medical researchers not to research the junk DNA?
That's what we heard today. That's what Sal Cordova told us.
Did he really say that? Because that's bonkers.
Yep, that's what he said. Evolutionists are not allowing the medical community to look into that junk DNA and find out that it really is doing something. So he said that
how wonderful it is.
We covered that video on the weather report, and yes, we have him saying that all loud.
That's bonkers. That's bonkers. I mean, the thing is, here's the thing. Here's the thing in science, and this kind of gets at the question of like changing ideas in science and going against a consensus. If you're the person who shows that a consensus is wrong, then you are going to win a Nobel Prize. That's how it works. For example, the guy who figured out the prions, Nobel Prize. The guy who figured out that a bacterium, Helicobacter pylori, causes stomach ulcers, when everyone thought it was not that. He proved it. He showed that that's the case. Like, I think he won a Nobel Prize for that. Ribozymes, Nobel Prize, right? You're not supposed to have RNA that's catalytic, but turns out that's a thing, right? If you can show that the consensus is wrong in a significant way, like, that's huge. Nobody is nobody does research to show that what we already think is correct is actually correct. The point of research is to find something new. It's Just a Walking Fish in the chat earlier said, the point of science is to make a pimple. And that is a reference to a description of what you do with a science education, where you get your undergraduate degree, then you get a graduate degree, like a master's, and then you're filling in your knowledge relative to what everybody knows. And then with a PhD, what you're doing is your knowledge gets right up to that boundary of what everyone knows, and then you pop it out just a little bit. You push on that boundary just a little bit, and you make a little bump in that boundary, like a pimple, right? And like, that is the point of science. If everyone is just going back over the same stuff and nobody is disagreeing, then everyone's wasting everyone else's time and their own. No one does that, because that's not how you make a career in science. You make a career in science by saying, hi, everybody, the consensus is wrong. Here's what's right. And here are the goods that prove it, right? That's how you make a career in science. That's what everyone is doing.
Right. So Redefine Living in the chat says he has a paper that says it's 98% junk. Produce the paper. We'll take a look at it. We'll see what they were talking about. We'll take a look at the year of the paper.
I bet that paper is from before 1990.
I bet it is. So
I
anyway,
bet that paper is from before 1990.
This illustration I have up is from the latest version of Molecular Cell Biology, which I think came out in 2016 or something like that. And they talk about the breakdown of what, okay, over here, this little red thing, I don't know if you see the red thing on the end, that is the protein coding gene, and that is the actual coded, the part of the genome that we could say is a code. And then over here we have introns, which are absolutely laced with non-conserved sequences, even though parts of them are conserved. And then, the plot gets thicker and thicker, but then, let's say then we have non-repetitive DNA that is neither in introns nor codons. And some of that stuff is transcribed. A lot of it's transcribed. A lot of it, we don't know what it does. There's a lot of probably pseudogenes. I don't know if the pseudogenes are included in the red part or not, but that's about 2%.
No, pseudogenes are not included in the protein coding regions because a pseudogene by definition is no longer protein coding. It doesn't get translated.
Whoops. Oh, and I just blew okay, there we go. So the green part would include the pseudogenes, and then over here on the left, we have all these repeated sequences. Now, repeated sequences, you got to ask, why are there millions of repeats? When one would do, right?
Because it's what they do. It's literally what they've been selected to do.
Right. Here's where all the viruses are, the lines and signs and ERVs and all of that. And this sort of gives you a breakdown of what's in the genome. It would be hard to make a case for all of that stuff particularly on the left, and even the stuff in the green version to absolutely be functional, because we know that even a variation between human beings, it is not completely conserved, and we can tell. And these human beings are all walking around. In fact, they can play tennis with each other and give each other a run for their money with these variations. So anyway, that's my point.
Just a short point on this, just to go through like what we know about functional stuff. So that little sliver of red on the right, that's functional. Everybody agrees on that. Within introns, you have teensy little regions at the ends of introns that basically say cut here.
The pivots.
Those are functional. Obviously, they say cut here to get rid of the intron. The rest of the intron.
There's a pivot in the middle too. So somewhere.
Yeah.
Yeah. Okay.
So you've got but an intron might be like 10,000 nucleotides, and then you'll have like a couple hundred at either end, and then in the middle, and the rest of it's nothing. And then within that green region, the non-repetitive, that's neither introns nor codons, some little bit of that is going to be regulatory. You've got promoters, silencers, enhancers. That gets you like a couple of percentage points in the genome, maybe, like being generous, 1 or 2% of the genome. You've got your repeats could be construed as functional if the number of repeats matters. So you could have situations where between element A and element B, you need to have 1,500 bases, but if it's 1,600 or 1,400, it doesn't work. So you could construe that spacer region, as, I shouldn't say spacer because that means a specific thing, but that kind of that in-between region, you could construe that as functional. You've got also repeats, centromeres and telomeres are largely repeats. Those are structural in nature. Yes. Those are functional. Everyone agrees. And then even once you get into the transposable stuff, whether it's signs or lines or retroviruses, whatever insertions you're looking at, some of those promoters have been co-opted. Okay, the promoters are like teensy tiny fractions of those, and most of them don't have the promoters anymore. The promoters have degraded away. So within, for example, the 8% of the genome that's endogenous retroviruses, only a fraction of a percentage, only out of that 8%, less than 1%, so less than 1/8 of it, are intact enough to even have usable promoters. And of those, a teensy tiny fraction of those promoters might be usable by like human genes, right? Might actually have a function for regulating human genes. So you go through these 20% this, 15% this, 8% this, 20% that. And sure, you could pick out 1% here, 1.5% there, maybe 2% there that's functional. But for every 1% you pick up, you exclude another 10 or 15% from being functional. Most of the genome at this point is not unknown. We know what it is, we know what it does, and we know how it got there. So like, I think everyone should continue to investigate exactly what every nucleotide in the genome is doing and why it matters. Because it is an interesting question. Why are there 3 billion instead of 2 or 4 or 10 or 1, right? That's an interesting question. We should figure out why. But like, we shouldn't pretend that we don't know that most of this stuff is non-functional. Most of it's non-functional, and that's it's not really up for debate, except in mostly creationist corners at this point. Even the ENCODE team has abandoned the idea of like 80% functionality.
They don't use the word functional anymore because they got a little bit of pushback on that.
Anyway, a little bit.
But even some of these long, these repeated sequences, they simply serve as rope to get from one place in the chromatin to another. And so there's some of that, and then there's many complexities. Like in these introns, there are actually some RNAs that code in the reverse direction that can actually repress the gene, or express the gene. And there's that. Cool. And then sometimes these lines and signs end up right smack in the middle of a gene and they either kill you, and there we have negative selection, or they stick around and they end up doing something in the future. So there are many complexities, and nobody is denying that, and nobody is telling researchers to stop looking. For God's sake,
that's bonkers. No one's saying that.
I mean, every day you could read a paper that will show you some new thing about DNA that you didn't know before. So
yeah.
Anyway.
It's no one's saying don't look at this, don't research it. That's silly. No one's doing that.
Yeah,
and
absolutely.
honestly, that's disappointing to hear that, because Sal knows better, I think.
Yeah. In the future, when we get to these repeated units and these lines and signs and ALUs and all of that, this is a very nice paper, 10 things you need to know about transposons, I believe it's called. And it gives you a nice little classification diagram here that kind of shows you how we classify them. And it has its own phylogeny, and they've done some remarkable things like this, where they've created a phylogeny of transposons, different classes and stuff. But we can get to that later. But anyway, hand it back to you, Andrew.
Okay. So I guess should I move on to part 3 of the questions?
Sure.
All right. So this one, part 3, is revival. I'm kind of gonna merge part of the questions for part 2 and part 3, just because I don't want to take up too much of the time. So this question is, why is science only based on things we can observe? And just to add on to that, how does that make science work so well, like today?
So that gets at the idea of methodological naturalism. Basically, if we're going to propose explanations for things that we observe, we need to be able to evaluate those proposals. We need to be able to evaluate the mechanisms. And this is distinct from philosophical naturalism, right? Philosophical naturalism is saying the observable, measurable world, universe, whatever word you want to use, is all there is. Methodological naturalism just says that in terms of answering questions scientifically, we are restricted in our investigations to things we can detect and measure. We cannot deal with explanations that are supernatural because we can't measure them, we can't quantify them, we can't detect them. And you get a lot of pushback on this idea by people saying, well, that's unfair, right? Because that excludes a whole bunch of potential explanations. No, it doesn't. If you have a potential explanation that you think is right, tell us how to test it. That's what it means. It means any proposed idea needs to be testable. And if you can't test it, it's not science. And
well, you can think about it and then get a burning sensation in your heart that tells you that what you were thinking was true. That's one way to test it, right? No comment. You can say no comment.
I'm going to say no comment on that one. Yeah, no, you have to be able to, right? You need to be able to measure things, right? You get this all the time with intelligent design is like, okay, you say structure XYZ was intelligently designed by a supernatural agent. How do I evaluate that? Like what experiment can I do?
Yeah, if Merlin did it here, right? How do you show it in the present that Merlin did it?
Yeah.
Right? So, and let's not pull any punches. We're talking about Merlin doing it with a magic wand. So if Merlin did it, and we have to keep an open mind, because now how would we find out in the present right here, in the near future, this is kind of where we are in science, right? So basically, how would we find out that Merlin did it? How would we figure that out?
I prefer Thanos and the Infinity Stones, but I get what you're saying, Speed.
Merlin is easier for me to remember.
Anything that these proposals come down to saying natural processes can't do X, therefore supernatural processes did X, right?
Yes.
And you know what? Okay, then tell me how to measure that. Tell me how to detect that, right? And if the answer is then it's not, then don't waste my time. Then it's not science. You can believe it all you want. You could say that it's the case, but like, if I can't test it, then I don't know what to tell you.
If you can show that it's something happened that is absolutely impossible, like out here in the present, if you could prove impossibility, then you might have something. But what they do is that's where they use this abduction. That's where that comes up. They'll say, "Oh, well, it's most probably impossible."
Too improbable, right?
Too improbable, yeah, that sort of
thing. And then you get into silly arguments that people have been having for 500 years, like, well, who designed the designer? It's like, now it's just philosophy, and like, I'm going to go take a nap now because this is boring. Like, get back to me when we're talking science again.
Yeah,
you just go down these silly philosophical rabbit holes, and the point is, once you go down a silly philosophical rabbit hole like that, you're no longer learning about how nature works. You've given up on trying to figure stuff out at that point.
And my main critique of the creationist camp is they got all this money, they're doing all this research. What they need to start concentrating on is proving what is impossible, for sure. I mean, prove to us something that's impossible by chemistry.
I feel like they've been trying, though. It's, and at this point, it really is just the origins or bust, because, they've tried irreducible complexity. They've tried microevolution versus macroevolution. They've tried information. And all these arguments just faceplant, because when you, look into them in any kind of systematic way, the evolutionary processes are fully capable of doing whatever the creationists say they are not capable of doing. So it ends up being abiogenesis all the time. It always reduces to that because, well, how did you get the first thing? And I mean, I understand why that's the case, because that's something we don't have an answer for yet. We're learning more and more every day. Literally just this week, there was a publication about tRNAs that is fascinating, and I need to do a deep dive because it's really cool, and it has to do with tRNAs and basically enzymatic reactions that they could be involved in, which is fascinating because we didn't know tRNAs could do that. And the tRNAs are also small, so if you have a random polymerization of RNA, tRNA-like structures are going to pop up. So, creationists would rather we just not do that, right? We just kind of throw up our hands and give up because it's too improbable, it could never happen, therefore God did it. Well, I would like to keep looking, personally. I would like to figure out what is and is not possible and see. Why should we're talking about how the scientific method works, right? And redefine living is illustrating the point, right? Abiogenesis is impossible. Done.
Yep, we're done.
I prove it. Show me that's the case. Show me why what is the thing, because not having an answer to something is not the same as there not being an answer, right? We do not know the pathway from no cellular life to cellular life. We have a bunch of hypotheses and have shown a bunch of the steps of how that could and would have happened, right? But we don't know the A to B to C to Z pathway, right? We don't know yet. We haven't figured it out. Creationists are saying, therefore we can't. Therefore there is no answer. It's impossible.
Yeah, we got to.
You know what else was impossible? Sequencing DNA, sequencing proteins, figuring out the rules of heredity. All of these things were impossible at some point. But we figured it out. And that sounds like a silly comparison now in the year 2021 because it's 2021, and of course we can sequence DNA, and of course we know how the genetic code works, and yada, 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 right? But in 1950, in 1940, in 1900, forget about figuring out the DNA code, just mapping the codons. They didn't even know that DNA was the thing that transmitted genes until the 1950s, right? We didn't figure that out until the Hershey and Chase experiment, which I believe was published in 1950, I want to say 52, right? Like really recently, we didn't figure this stuff out, and it didn't become the consensus. So I don't want to hear, in science, saying something is impossible is a non-starter. It's just not part of the process. You don't do it. You try. Get caught trying. And honestly, creationists, you have a couple of think tanks that have bajillions of dollars, right? More money than God, some of these creation research groups. You know what? Figure it out. Get in the lab, and you do your research instead of throwing stones at the people who are busy actually figuring stuff out.
Right.
Yeah, and an interesting thing that just popped up, and let me take a minute to explain this. Basically, we have eukaryotic cells with the nucleus right here, right? And we have these pores, and there's a guy on the internet who we reviewed. We reviewed one of his videos earlier, but he shall go nameless. But he had a theory. He said, "How could this possibly have evolved? How could we have these nuclear? Okay, each of the things that are transported into the nucleus have something called a nuclear localization signal." And things that are transported out of the nucleus have a nuclear export signal. I think it's an NES. Might have a different name. I'm not sure. He said, how could those things have evolved? Well, when I looked into it a little bit, I found a paper that, lo and behold, in prokaryotes who do not have a nucleus, it turns out that some of the ribosome machinery is made with the and I blew my little drawing. It is made with these NLS signals. They're in the ribosomes of prokaryotes that don't have a nucleus. So we have nuclear localization signals, but no nucleus. This is prior to the nucleus. So because we looked, we found out that this goes back possibly way before the prokaryotic eukaryotic, marriage or whatever it was. So anyway, that's an example of how, if we had just given up, why would we have ever looked for that commonality? That's
Right.
Anyway, that's my point.
And
So
go ahead.
Oh no, or go ahead.
I was just going to say a related idea to that is the idea of irreducible complexity of, that what Speed just basically described was this system is irreducibly complex. How could it have evolved? Well, let's get caught trying to figure it out instead of throwing up our hands and saying it can't.
Right.
And lo and behold, we figured it out, right?
RJ is here. RJ Downard is here, and Immutable is in the room. He's
Hey! I know. I persist. I don't know. It might be that I have access to your StreamYard.
How did you do that? You hacker!
Damn,
hacker.
I wonder. Well, you consented to it a few months ago, so I remain innocent of those charges.
Yeah, I haven't figured out how to take it away yet. So,
what's today your mall?
Hey, shut up.
Okay.
Because you socialize in, right? That's your social
life. Yeah, man.
I just wanted to get your social life because I know personally how important it is not to be isolated.
Look,
to be more like an old married couple.
I try to stay like as far away from home as possible because it usually drives me insane doing it.
Yeah.
For too long. But anyway, Dr. Dan, eukaryotes undergo vertical gene transfer, don't they? With other eukaryotes occasionally? Or I'm mistaken.
You mean horizontal?
No, it's something.
So vertical gene transfer is just parent to offspring.
Yeah.
Everybody does that.
But
horizontal is the weird.
Do eukaryotes undergo gene transfer with other organisms?
Between each other. Well, between
all the time.
Prokaryotes.
Okay.
Do they?
I was meaning to ask, if you consider the last eukaryotic common ancestor before that, is it known or is it speculated that gene transfer would have been a large part in the development of eukaryotes, or is it
huge part? Yeah, because a lot of the major events in the development of eukaryotes, right, would have been nucleus and mitochondria are the two, eukaryotes. We do a lot of things that are the same in terms of things like translation is the same across all eukaryotes. The membrane structures are very similar across eukaryotes and distinct from prokaryotic membranes. But two of the big things that structurally, right, that eukaryotes have, it's nucleus and mitochondria. And mitochondria certainly were endosymbiotic, and I actually don't have a position on the origins of the nucleus in terms of whether that represents an endosymbiotic event or not, but I don't think we should rule it out. And endosymbiosis is all about horizontal gene transfer. It's all about exchanging protein coding and regulatory DNA sequences between compartments, right? And we know that happens in the present, because we've got, for example, animals that have genes for photosynthesis that they've acquired from their green algal food, that they practice something called kleptoplasty, where they steal the chloroplast from their green algae food and they run on kind of backup solar for, depending on the organism, days to weeks. And the ones that have the long-term associations are able to do that because they break down the algal cells, they keep the chloroplast, they keep the plastids, but they have incorporated some of the algal genes from the nuclear genome of the green algae that code for genes that are trafficked to the plastids that are required for photosynthesis. So the one I'm thinking of, it's a marine organism called Alesia chlorotica is the one I'm thinking of, has a gene for photosynthesis in its nuclear genome. It is people are going to skewer me because I don't know it. I forget if it's a flatworm or a mollusk, honestly. I think it's a gastropod, but don't quote. I'll look it up real fast. But it has acquired genes, so it's an animal that has genes for photosynthesis that it has no business having, but it's because it uses the chloroplast from its food to do photosynthesis.
Yeah.
Like, so we know that this happens all the time.
I accept endosymbiosis as an event that occurred. I mean, I know there's some people positing that it might have been like parasitosis or symbiogenesis that caused, eukaryotes to really fully become a thing. But
it's a gastropod, by the way. I just looked it up. It's a gastropod. It's a mollusk.
Okay. But would it be safe to argue that if it was just like symbio or endosymbiosis, that other prokaryotes would have also taken the eukaryotic trajectory, or like is there something like notable to our symbiotic event that would have really kind of segregated us from the rest of, at that point, like proto-eukaryotic life?
Yeah, that's a really interesting question, because genetically we can pretty conclusively say there's a last universal, or a last eukaryotic common ancestor, right? There was one population that all the major groups of eukaryotes are descended from.
Right.
An open question is it the only time that happened, or did it happen multiple times and the ones that are left are the ones that won? And I don't think we can rule that out. I think that's a really interesting idea.
I think it's a question of like significance. I mean, certainly it's significant. Like, that's not to be like challenged. But is it the most significant event? Like, is that the hallmark event, like that really segregates?
Oh, for eukaryotes and prokaryotes?
Yeah, like that segregates like eukaryotes from like prokaryotes and archaea.
I think in terms of the extant groups, yes. Because we don't know of any eukaryotes we don't know of any organisms that have a archaeal cell morphology, right, that nest within eukarya. Does that make sense?
Yeah, but aren't archaeal like cell wall structures and eukaryotic cell wall structures like remarkably similar to each other?
They cell walls, I don't know. The membranes are more similar. I don't know about the cell walls. I know that nobody uses chitin except for fungi. I don't think anybody uses cellulose except for the archaeplastids, but I'm not 100% sure about that. I don't think so, though. I don't know enough about archaeal cell walls to say for sure. But in terms of the membranes, there are kind of three, again, cellular biologists know this more than I would, but my understanding is there are kind of three distinct groups of membranes. There's the bacterial membranes in terms of their like phospholipid makeup, and then there's archaeal membranes and eukaryotic membranes, and those two are distinct but more similar to each other.
So most bacteria tend to use NAM as part of their cell wall structure, right? NAM being N-acetylmurambic acid.
I couldn't tell you.
It's also called like peptidoglycan.
Oh, peptidoglycan, that's a word I know. Yes, bacteria do that, and nobody else does. Yes, yes. Right.
So I had to, watching chat a little bit, Redefine Living is one of our critics, and he came up with this thing that, what was it that they showed that you have to have, James Tour showed that you have to have absolutely pure chemicals to do things, right? And what that shows you is that purity works, right? And I'll get rid of that. Purity works. Now, hang on a second. How does that prove that an impure system doesn't work. It doesn't.
Well, the thing is, as well, is like, why is it not possible for an impure system to tend towards like an abundance of some molecules over others, right? Because the entire point of like the very early phase, like of abiogenesis, is that you have multiple different environments that are changing, and some molecules that are stable in one environment but not stable in the other. And so, like a lot of chemicals that might be like sensitive, like to photosensitive reactions, like photolysis or something like that, would degrade in an environment like where they're exposed to UV radiation. But if the environment changes to such that like it's like the environment is no longer, or and those molecules are no longer like exposed to UV light consistently, well, then that's not an issue, and photosensitive molecules that are remaining would remain in that environment as long as that environment persists.
Yeah, I mean, but basically, just because we use purified chemicals in some sort of research project, if you did not use purified chemicals, you would never know what happened. If something did happen, you would have no way to know what happened if you use a bunch of stuff. But what we're doing is we have trillions of little test tubes, basically, trillions, quadrillions, quintillions of test tubes in prebiotic Earth, and we're just trying to find the pathways. So we're looking for this one path to the next path. And in order to find that path, every one of these is a experiment done with pure chemicals. And they don't seem to understand that that's how you got to do it.
And this gets at an important point, that the purity is required to figure out what happened, because you are testing for specific things. You're testing specific predictions. But in the abiotic environment, they say you need everything to be pure or it doesn't work. No. If it was all pure, it would not have worked, for the specific reason that Immutable just explained. If it was an environment that was very uniform with all pure chemicals of whatever it was, it would not have worked. The reason it works is the diversity. I do want to get back to Andrew's stuff. I want to make sure we cover all his questions, because we should leave a gap between this and Snatta.
Yep. We need a gap because I need to make another cup of coffee. So go ahead, Andrew.
Okay. So I just had one more question for you, Dr. Dan, from Heart for Modernity. I cannot speak.
Modernity?
Yes. Thank you. I can speak English. So I think this is probably a really relevant question in terms of today's scientific community, basically, or just the YouTube community, I guess I should say. So Dr. Dan, I'd like to ask you, how has the internet and media affected science and public trust in it?
Oh, that's a good question. It's such a double-edged sword, right? Because it's so much easier to get information out, but it's also easier to get incomplete information out. You look at, for example, there are a bunch of science websites that basically will summarize research findings and blast out the most hyperbolic headlines possible, and then people see the headlines without reading the paper, and it's a mess. I think it really does, as in many things, not just science, but it really does represent a missed opportunity to do a good job of conveying accurate information to the public. And on the other side of that, it opens up such an opportunity for misinformation to propagate. I want to say on net it's a good thing, right? Because I get so mad about the barriers that science puts up that prevent non-scientists from understanding.
Yeah.
It really frustrates me when scientists deliberately make it hard to access science. And a lot of that is not scientists. I shouldn't say scientists, because if you email, by the way, it's just a tip for everybody. If there's a paper you want to read and you go on, NCBI or something, and you can't access the full text of that paper, email the lead author. Guarantee you that person will email you a PDF of the paper. 100%. That's how you get papers if you can't access them for free, legally. So it's not, the barriers are not from scientists. The barriers are largely from the publishers. And now there's a lot of really good open source publishing, both in terms of like primary literature and textbooks, which is great. YouTube. But again, it's a double-edged sword, right? So you also have the problem of communication, and scientists sometimes not the best communicators for a non-scientific audience. So if you have somebody that is a better communicator peddling BS, that will often be trusted more than a scientist providing accurate information in a less accessible way. And it's something that scientists really need to work on. I really think that front and center in a science education needs to be science communication. It really is critical because, I mean, just look at where we are right now with the coronavirus, right? You've got what percentage of the population still think this is made up. You've got what percentage of the population think that the vaccines are Bill Gates that are going to try to control your brain, right? The science is out there, the correct information is out there, and it doesn't take a lot of effort to find it. It's just who is doing the best job getting their claims out there, and the answer is not scientists.
Right.
And that's the problem.
We don't want to be ruling things out because ever since Scott got the vaccine, his Spotify has been like right on. I mean, it's just been like
nice.
Yeah.
That's an issue.
But it's frightening. It's frightening.
Yes.
Let's see. Redefine Living made an interesting point a few moments ago.
Because if life was this one, the one I have up.
Yeah, because if life was simply in the assembly of in the arrangement of a cell, they would never die.
Oh my God.
Dr. Dan, my perspective on life is kind of maybe it's too elementary, but I tend to think of life as not being like definable in terms of like a number of ingredients, but more about a process.
Well, just think about the logic of what he said here. Does that mean the cell never changes once you assemble it?
Well, on the contrary. I mean, cells are continuously changing. I mean, various groups, like molecules that are in a cell, are undergoing chemical transformations to become carbon, or to be converted into citrate or into glucose, or all sorts of things that a cell needs in order to survive. I mean, a cell. There is nothing static about a cell. It's always changing, and it's always, its components are always being used in some way based upon the need of that cell.
Stephanie Trenton just said something brilliant here.
You must have.
So this is one of my favorite questions in biology is, what exactly does qualify something as living? And you could make the argument that, like, Dan, why do you think that's an interesting question? You tend to not like this philosophy stuff. Well, I think this is actually an empirical question, but the answer is not that there are a set of characteristics that make something living. It's that it's a continuum of increasing degrees of biological activity. So most cells are larger and more complex and do more biochemical things than most viruses. But there's overlap where the smallest, simplest cells are smaller and do fewer things than the largest, most complex viruses. On the other end of that, the smallest, simplest viruses work basically the same way as the largest, most complex ones. They just have their host cells do everything for them. And there are plasmids and there are transposons and there are integrating elements that do more stuff and are more complex than those simple viruses. And you keep going down through, viroids and prions and everything, and all these different biochemically or biologically active entities that circulate within and among cellular life. Where do you draw the line on that group between living and non-living? There isn't a place to draw a line.
So, I mean, with the definition you're giving, I think I know some programmers who would say that they can design like codes or systems that behave very similarly to biological cells in the sense that they change based upon their environment and things like that. Although perhaps we should be cautious or kind of be a little more careful in our definition of life in saying that a cell appears to have some kind of quality of being fundamentally like non-deterministic.
Well, let's say it has to be a physical medium, a physical thing that does this stuff, because then that excludes like computer programs.
Well, physical mediums are still responsible in computer programs, right? I mean,
Yeah,
you have electrical.
Like you can't
circuits that
isolate the DNA from a computer program. Like there's no molecule that is the DNA of a computer program. The ones and zeros at the end of the day are not physical entities.
Yeah, did you know there are no ones and zeros in computers? Actually, not really. They're
I mean,
it's the days of punch cards.
They're little. Okay, they're not square waves. They're little rounded waves, and you have to catch them at the right moment in order to read a 1 and a 0 out of them accurately. So, I mean, even if you look at an actual computer program running, it's pretty analog. But
I mean, it's curious because there are people that are wanting to do transition into what they're calling molecular robotics. I'll send you a music video on it, which is actually what introduced me to it. But it's a system where they're exploiting like the chemistry of specific systems to essentially make it like programmable matter. It's fascinating. It's way beyond my pay grade.
Yeah. But at that point, it definitely gets weird, but we're not there yet. So thankfully, we don't have to grapple with that.
Yeah.
No, but I mean, would you say, like, for instance, cells seem to like drastically change their behavior based upon the environment like you put them in? Like if you stop giving a cell, for instance, like food or the nutrients it needs to survive, it'll start doing something very different. Like it'll start conserving more energy and it'll become more dormant in its activity, usually.
So, Andrew, is this everything that we promised? Do you have anything else?
Oh, well, I guess I just had like an unrelated question, but I'm not sure if Dr. Dan has time or if it's kind of related to the subject we've been discussing so far.
Up to you guys.
I think if it's phylogenetics, I think save that for another time because this has kind of been a fairly targeted discussion on kind of the underpinnings of science. So trust me, I could go on and on about phylogenetics, and I would love to do that at some point too.
Yeah, and I'd like to at some point get into retroviruses a little bit and start looking at the classification and maybe try to explain some of that and address the idea that they cannot be co-opted into some evolutionary pathway because they've been around for what, 190 million years.
You had me at viruses.
What's that?
You had me at viruses. I'm
okay.
in.
Yeah, you like viruses. Okay. Anyway,
yeah, look,
I have a whole bunch of illustrations to help us with that task. So anyway,
cool.
I like pictures.
Dr. Dan, fun fact: did you know that they discovered viruses that infect other viruses?
Oh, yes.
Little devils.
Virophages. Yeah, the coolest. It's incredible.
What's the deal with that anyway? I didn't think they were supposed to.
That reminds me.
They're not. Yeah, they're not supposed to. This gets into a whole thing about are the big viruses viruses or are they intracellular parasites that transmit via viral-like particles? Yeah. It's an open question, but it's well beyond the scope of what we're talking about tonight. But don't get me started on those giant the NCLDVs and the virophages. Like, do not get me started because we will be here until tomorrow.
That kind of reminds me of the Christians attacking each other on Praise's channel.
It took me a minute, but I got it. It took me a minute.
Yeah, I want to learn more about viruses because now that I've heard like virologists talk about viruses and some of the quirky little behaviors they exhibit, it's like, wow, these fuckers are really complicated.
Let's do it sometime. It's super fun. It's incredibly cool and weird. And viruses, there are no rules. The first rule of viruses is there are no rules. They can do whatever they want.
But the really weird.
Go ahead.
All right, but let's save viruses for another time, though.
Okay. Yeah, I will. I could just talk about this ad nauseum.
Can we wrap up then, guys?
I guess so. You old ass man.
Oh, you young little weenies. I don't know. Okay, I take so much crap from these guys. Okay,
one of these
days, mutable.
One of these days.
Next time.
Thanks.
Thanks,
Speed. This was fun, and thank you, Andrew.
Yeah, we need to do more sciencey stuff. Damn it!
Yeah,
I know. I'm trying. Right? I know.
And so is Andrew. like
we could have a section where Dr. Dan teaches Immutable about viruses, because as a biochemist, like, you don't really learn too much about them. It's like, yeah, these are a thing.
Yeah,
Back to cell.
We could do that.
The problem is that Andrew has set up many things. I'm such a softie. When somebody asks to be let in, I let them in, and then I have 10 people, and then it just all goes to hell. Somebody has to get control of me. So,
Hey,
I've been trying to for like a year now, and that's been less than a fruitful endeavor.
Keep working on it. Okay, we're going undead, guys. So anybody have anything?
We're having Sonata at 9, right?
Sonata at 9. So
that's
going
in
undead.
45 minutes.
Goodbye.
It's 9 Central.
Goodbye, curious people.
Okay.
What Dan said, Central.